So the process of DNA replication is shown here. This goes all the way back to the story about Watson and Crick and the discovery of the structure of DNA. So already when they came up with the model of the double helix, the two helical strands wound around one another, they were thinking about the process of DNA replication. And you now know everything you need to know to be ready for an understanding of DNA replication. We've taught you that in previous modules and in previous objectives in this module. Recall that there's a very specific base pairing rule, that A always pairs to T, that T always pairs to A, that G always pairs to C, that C always pairs to G. And so in that way, we can make a copy of the strands if we only pull them apart. If we pull those two strands so that their backbones are separate and they're sitting apart from one another, now we've got what's called single-stranded DNA. We can see near the middle of this diagram that the base A, or adenine, is being paired up with the base thymidine, or thymine. And that's where the tritiated thymidine came in, is this yellow hexagon with the T on it is what we were adding as the raw materials, and how we knew that we were adding those during the experiments that we were doing that I described in a previous video. So in that way we're building the new strand that's shown here with a teal backbone. The old strand had a purple backbone. The new strand has a teal backbone. So that teal backbone, or light blue color if you're male and you don't do colors or color names that way, the light blue or teal backbone is showing you the new strand. Remember I said earlier that everything has to be built in a 5' to 3' direction. And that's going to be an important part of this next story. And the next story is about something called an Okazaki fragment. So DNA is anti-parallel when it's a double helix. We looked at that earlier in this module. If the structure of DNA and the idea of two anti-parallel strands is unfamiliar to you, then you'll want to go back and review the video or the study guide materials for Module 5, Objective 3, and you'll see those terms discussed there. But that's an important part of what we need to understand the process of DNA replication. So because those strands are anti-parallel always, and because we always build in a 5' to 3' direction, then for the top strand that's shown here, what's called the leading strand, then that's an easy thing to do, because the blue strand is running 3' to 5', and we're building off of it 5' to 3', and so that's no problem at all. We just build our way along 5' to 3'. We keep adding new subunits. We keep adding new thymidine or adenosine or cytosine. So we keep adding the bases with their deoxyribose sugars attached along and healing it and sealing that together along the phosphate bonds. And we just keep doing that over and over again in a dehydration synthesis reaction. And we do that over and over again at that arrowhead that's shown at the 3' end of this leading strand. This is a relatively easy process. The problem that we have then is on this bottom strand shown in this diagram, the lagging strand, and we use Okazaki fragments to solve this problem. Remember that we've got to build an anti-parallel strand, and that build always has to go in a 5' to 3' direction, in the direction of the arrows that are shown here. In this bottom strand, the parent that we're using as the template runs in the 5' to 3' direction, and so we can't just simply copy it, because we've got to copy it in an anti-parallel direction. Our two arrows, in other words, have to run in opposite directions when we're making the copy. So what we do is we make little short segments that are called Okazaki fragments. These are little short pieces of DNA that are built off of an RNA template in a process that you don't need to fully understand at this point in your career. And those Okazaki fragments in humans are probably about 100 to 200 base pairs, 100 to 200 nucleotides long. And so this arrow that we see here, the blue arrow, is representing about 100 to 200 nucleotides. And these short segments of DNA, which are built in a 5' to 3' direction and then stitched together, are called Okazaki fragments. As you might imagine, the process of building the leading strand and the process of building the lagging strand can't occur at the same rate. The leading strand is built much faster than the lagging strand. This creates some special problems for the cell that we don't need to get into, but it's something that you might want to be aware of.